Hello, students, and welcome to another class for Environmental Biotechnology. We will be dealing with lecture week 4, assignment related questions from lecture week set 4. I hope all of you have some time to go through your lectures. And every Saturday, 3 p.m. to 5 p.m., we have the time designated for this live session. And my name is Ashutosh, and I am the teaching assistant for this course. This course is taught by Professor Pinaki Sar, who is from IIT Kharagpur, and I am a PMRF PhD scholar at IIT Kanpur. So I see the population is thin today, but no issues. This will be this will be a recorded meeting, and this will be uploaded on YouTube. The recordings of this, but I would encourage everyone to attend the live session and clear all your doubts. Move along with me and solve these questions so that you get a first-hand experience of how what kind of questions you will be facing in your exams as well as in the assignments. This will also serve as a mode for revision for all of you to touch up on these topics if you are not able to follow the lecture series. Okay, and the format in every class I will be repeating the format of this presentation. I have designed 10 to 11 questions for all of you guys, which are multiple choice questions which will be solving in this 2 hour session. And after each question is posed on the screen in the presentation, I will wait for all the participants and those who will be watching this video for some time so that they can think about what is the correct answer. You can communicate your correct answer to me through the chat box. I'll be following the chat box regularly, and then after a minute or so I will reveal what is the correct answer, and then we will understand the logic behind why the correct answer is the chosen correct answer. Okay. Meanwhile, we will start this class. Anyone who is requesting to join this link will be allowed in, and I would request all the participants to please turn off your mic and video chat for the smooth conduction of this session. Only my video and mic would be turned on, and I will be speaking throughout this session. Please communicate with me via the chat box, which I'll be following very closely. Okay, with that much of an introduction, let me share my slides, and let us begin this session. Can you see my slides, students? Is my voice clear and audible, and can you see my slides? Just let me know in the chat box with a yes or no, then we can begin. No, sir. Okay. I think I have to reshare my screen. No. Maybe I have to share my window. Let me reshare my screen again. Okay, I think it should be visible now. I have changed the settings and reshared my screen. Can anyone let me know in the chat? Can you see my slides, and can you see the questions? Yes. Thank you, Priscilla. Okay, so let us begin this session. Week 4 of Environmental Biotechnology live TA session, and my name is Ashutosh. I'm a PMRF scholar at IIT Kanpur. So, and this course is brought to you by Professor Pinaki Sar and by NPTL. So, NPTL authorities will be sharing the links for this recorded session as well as the presentations that I'm going to present here. All the example questions will be provided to you as materials. Okay, this is the format. This is the same format that we will be following from the previous classes. I have prepared 10 example questions from lecture week 4, all the lectures that Professor Pinaki Sar has taken, recorded in lecture week 4, and we'll be dealing with them one by one. Multiple choice questions in which there are multiple options, only one is the correct option. Let me know in the chat box what do you think is the correct option, and then we will understand the concept behind the correct option. Okay, so moving on to the first question of this session. Question number 1: total DNA based metagenomic approaches are not adequate to gain a complete understanding of a microbial community because. So I hope you understand the question. This question is telling us about what could be the possible limitation of metagenomic approach which is not adequate to gain a complete understanding of the microbial community. Option A: all genes cannot be detected. Option B: unculturable microorganisms cannot be detected. Option C: all genes are not expressed. Option D: all metabolites cannot be tracked back to the genes. And option E: all genes are not protein coding. So these are all the options. Total DNA based metagenomic approaches are not adequate to gain a complete understanding of a microbial community because dashed. Let us take some time, a minute or so, and I would request everyone who is present in the live class as well as people who will be watching this video, let me know your answer in the chat box. Think about it, and I will come back to this question after a minute. Okay, I think that's enough time for this small question. Total DNA based metagenomic approaches are not adequate to gain a complete understanding of a microbial community because. Let us see if someone has answered in the chat box. Ashwini has answered option C. Let me reveal what is the correct answer to everyone. The correct answer to this question is option C. All genes are not expressed. And this is one of the limitations of DNA based metagenomic approaches. Well done, Ashwin, you have answered correctly. Let us see what are these limitations of metagenomic approach option by option. Okay. Now, option A and B, all genes cannot be detected. Now, in a metagenomic approach, this is partially true, but this is not the main reason why we cannot detect the composition of complete understanding of a microbial community, because some low abundance microbes might be underrepresented, but sequencing depth can often capture most genes. This is more of a technical limitation, not a fundamental one, such as rare species in soil microbiome may be missed if sequencing coverage is shallow. Okay, so this is a technical difficulty of metagenomic approach, that in a microbial community or in a biological sample which is undergoing metagenomic procedure, there might be some low abundance microorganisms. They are present in very low number. But it depends on the sequencer and the depth of the sequencing. If the sequencing depth is very high and the coverage is good, then it can also pick up low abundance organisms. So I have mentioned here the concepts of breadth and depth of a sequencing. Depth is, when I say depth of a transcriptomic or a genomic sequence, we mean, what do we mean by how many reads and contigs align to the reference relative to the reference sequence length. So how many number of contigs can be aligned to a reference sequence that comprises the depth of the sequencing. This unit does not account for uneven distribution and contigs over the reference sequence length. How deep or how good is your sequencing length? What how many number of reads can be mapped onto the reference genome? That is the depth. And what is breadth? What percentage of the reference genome length is covered by reads and contigs? So this is breadth. Across the reference sequence, how many different contigs, your contigs from the sample can span, if they can span a larger length of the reference sequence, then that particular sample is said to have a good breadth. While the large number of reads in the same reference sequence is depth, while spanning the whole length of the reference sequence is breadth. So if a sequencing technique and a machine sequencer has good coverage of depth and breadth, then it can also pick up low abundance organisms. So this option is not the correct option, because this is only a technical limitation but not a fundamental limitations to metagenomics. Option B, unculturable microorganisms cannot be detected. So this is absolutely not the correct option, because the major difference between metagenomics and pure culture related culturomics has the advantage, because metagenomics does not need pure culture and can detect unculturable microbes as well, because we are simply lysing every cell and all the microbes present in the community and identifying them based on DNA sequencing. The main advantage of metagenomics is that it does detect unculturable microbes. This is what I was explaining. Since DNA is extracted directly from the environment without culturing, no pure culture needed, no subculture needed, you directly lyse the cells of the extracted microbes and just do the sequencing of the DNA to identify the organisms. So it does not needs culture and can also identify unculturable microbes. Example, 99% of ocean microbes are unculturable, yet metagenomics can still recover the DNA. So best example is a sample from the seawater or deep sea regions, which is not culturable in any media or in laboratory, but still metagenomics can pick up those organisms since it is just based on DNA sequencing. Now coming to the correct option, which is a fundamental limitation of metagenomics, all genes are not expressed, which is the closest or the most correct answer, because many genes may remain silent under given conditions. To understand activity, we need metatranscriptomics and metaproteomics as well. So just by DNA sequence, we cannot say that what is a function of the community, and many genes are not expressed under a given condition. Not all the genes are expressed under a given condition. Only a subset of genes are expressed. So all genes are not expressed is a valid limitation of metagenomics. Example, antibiotic resistant genes may be present in gut microbiota but not expressed unless antibiotics are introduced. That is a correct example. The gut microbiota, if you do a metagenomics, many of the antibiotic resistant genes will not be expressed since there is no antibiotic. Only in the presence of antibiotics such genes will be expressed, which might be missed in the metagenomics study. Therefore, it needs to be paired with metatranscriptomics, which studies the mRNA levels of the microbial community, and metaproteomics. So understanding these terms, this is the genome or the genomic DNA. The genome is consisted of DNA in the chromosomes. When genome gets transcribed, it turns into a messenger RNA or the transcript, and the study of messenger RNA is transcriptomics. And then these messenger RNA gets translated into functional proteins, and study of those is called proteomics. And then proteins act as enzymes and catalyze different reactions and products which form the metabolites, and the study of those is known as metabolomics. So in whole environmental biotechnology, metagenomics approach should be paired up with metatranscriptomics, metaproteomics and metabolomics. And the other two options, all metabolites cannot be traced back to genes. This is true, but this is a secondary limitation of a metagenomics, not a direct one. DNA based data doesn't tell us about the functional output directly, because as we saw this hierarchy, changes or sequence of the DNA will not say what are the metabolites generated in the community or inside a cell. Linking metabolites to gene requires metabolomics plus integrated multiomics. For example, gut microbes may produce butyrate, but metagenomics cannot confirm which strain is actively producing it. So this is a limitation, but this is a secondary limitation to metagenomics approach. All genes are not protein coding. This is also true because, as we just saw here, only messenger mRNA, messenger RNA or mRNA codes for proteins. There are other transcripts which arise from genome, such as long non-coding and circular RNA, which do not code for enzymatic proteins. DNA includes non-coding regions. While relevant, the bigger limitation is that metagenomics can't reveal activity or function. rRNA genes dominate metagenomes but do not directly reflect metabolic activity. So hence the fundamental limitation to metagenomics approach is because all the genes are not expressed at a particular given of time. And to overcome this, we need an integrated omics approach for transcriptomics, genomics and metaproteomics along with metabolomics to understand the community behavior in an environmental sample. I hope it is clear to the students. If yes, just let me know in the chat box and we will move on to the next question. This was a pretty easy question, and okay sir, okay, very good. Moving on to the second question of this session. Microbial matrices are dirty, making extraction of cellular material difficult. Dirty refers to dashed. Again, a question on metagenomics again. Microbial matrices are dirty, making extraction of cellular material difficult. Here, dirty refers to dashed. Option A, presence of interfering chemicals. Option B, presence of soil particles. Option C, presence of other microorganisms. Option D, presence of eukaryotes. Option E, presence of extracellular DNA from dead cells. Which of these is referred to here as dirty, which makes extraction of cellular material difficult while processing a sample for metagenomic study? Let us take a minute or so and let me know your answer in the chat box. I think it's enough time for this question. Question number 2. Microbial matrices are dirty, making extraction of cellular material difficult, and dirty refers to what? Let us see the chat box for the answers. Ashwini has answered option A. Okay. Let me reveal what is the correct answer to this question. Dirty here refers to option A, presence of interfering chemicals. Again, Ashwini has answered the correct one. Well done. Presence of interfering chemicals here refers to dirty in processing a sample for metagenomic study. Now let us understand what is the dirt exactly and what are these chemicals. Now, environmental samples like soil, feces, sediments, wastewater, which are often the samples for metagenomic study, often contains humic acids, polyphenols, bile salts, and heavy metals, as well as organic inhibitors, etc. So that is why from the soil sample or environmental sample, cells needs to be separated from this contaminated soil, and this contaminated soil, the, the cells that are separated then followed by lysis and DNA purification to then understand what are the different kind of bacteria and microbe community thriving in this contaminated soil. So this procedure is very tedious because the chemical contaminants are very difficult to get rid of, and soil the topsoil or humus contains humic acids and polyphenols which can interfere with many enzymes in the extraction process, in the PCR reactions, and the downstream processing for sequencing. These chemicals co-extract with DNA and RNA. So the way we extract separate DNA from a cell suspension is via making phenol chloroform isoamyl alcohol methods, in which the aqueous phase and hydrophobic phase will consist of most proteins, and aqueous phase is where DNA will be eluted out. So such presence of such chemicals interfere with the phase separations when treated with the DNA separation buffers, and also co-extract with DNA and RNA and interfere with PCR sequencing and enzymatic reactions. This is the main technical problem, and this is what is known as dirt in isolating microbes from the environmental matrices. Once the isolation is relatively pure, this soil DNA, the cells is lysed and the DNA is obtained. This soil DNA is cloned into various vectors and transformed into host cells, where the recombinant plasmid and such libraries are screened for the presence of different microbial communities, and the sequence is then mapped onto a reference genome. Now the other options that we saw, presence of soil particles, these are mostly physical substances and can complicate extraction, but they are usually removed by filtration or centrifugation, which is not the main issue. If you centrifuge the sample enough, then soil particles being heavier will settle as a pellet or in the bottom of the centrifugation tube, while cells and DNA being lighter will be in the aqueous supernatant. Soil grit may reduce yield but does not necessarily inhibit enzymes directly. Presence of other microorganisms, that is natural, because in any environmental sample there is always a group of millions of microorganisms. There might be many other microorganisms, and it is a complex mixture of such microbial communities. It is never a pure culture. That's expected. We want all the microbes in a metagenomic sample. Not a reason matrices are considered dirty. So soil, separation of cells from the soil to create a cell suspension, and then lysis, which is then followed by crude nucleic acid treatment with DNA and RNAs to, and then RNA and DNA purification methods. Presence of other eukaryotes. This is also true because there are animals in the rhizosphere. There are protozoans. There are helminthes and cysts which will be contaminated in the soil, but this does not interfere with the downstream PCM sequencing and assay. If some student has joined recently, I if I can request them to please turn off your mic and video chat while this class. You can interact with me via the chat box. So we are discussing a current question, question number 2, I guess, and you can join from question number 3. And if you have any doubt, please post it in the chat box. Yeah, as I was saying, presence of eukaryotes in the matrix for metagenomics. This is more about the background reads that will not be referenced to the reference genome. Not what dirty means. Example, human DNA in stool samples often overwhelms microbial DNA, but this is not a contamination which makes a sample dirty as such. It will all depend on the sequencing reads and the analysis, which can separate out human DNA from the bacterial and the microbe DNA. Presence of extracellular DNA from dead cells. Again, this is a different technical challenge, misrepresenting active versus inactive community, but it does not make extraction difficult as such, but makes interpretation difficult. Same as the presence of eukaryotes. Interpreting the sequence reads and data is what is difficult, but the dirt that we were referring to this question, which makes the extraction of cellular material difficult, is the interfering chemicals like polyphenols, humic acid, bile salts, heavy metals, and organic inhibitors. I hope everyone is clear on this question. Let me know in the chat box with a yes or no. The extraction process is difficult because there are chemical contaminants. Other biological contaminants make the interpretation process difficult. Okay, I received a response in the chat box. Thank you, Ashwini. Let's move on to the third question. Question number 3. Lithotrophy involves the utilization of dashed. Lithotrophy involves the utilization of dashed. Option A, organic matter as carbon source. Option B, inorganic matter as carbon source. Option C, lipids as carbon source. Option D, light as energy source. Option E, chemicals as energy source. If you remember the definition of lithotrophy, that is what is being asked in this question. Lithotrophy involves which of these characteristics? Let us take a minute or so and collect some answers in the chat box, as well as those who will be watching this recorded class. Please think about the question and write down your answer. Okay, I think it's enough time for this question. Lithotrophy involves the utilization of, and Saida and Ashwini both have answered option E. Now let me reveal what is the correct answer to this question. Lithotrophy involves the utilization of chemicals as an energy source. Option E is the correct answer. Well done, both of you, Saida and Ashwini. Chemicals as an energy source is used by lithotrophs, while organic matter as carbon source is used by organotrophs. And let us see what are the different characteristics of lithotroph. Lithotrophy or chemolithotrophy is a metabolic process where microorganisms obtain energy by oxidizing inorganic compounds. So it is the oxidation of inorganic compounds that gives them energy. That is what is characterizing these lithotrophs or chemolithotrophs. These organisms use chemicals as an energy source, often in environment where organic matter or light is scarce. So this happens in environment such as the aphotic zone or the anoxic zone where organic matter or light is scarce. So there are many microbes which are lithotrophs, use chemicals as an energy source. And one of the best examples of such microorganism is Nitrosomonas. Nitrosomonas is a Nitrosomonas is a nitrogen fixing bacteria which can oxidize ammonia. The bacteria Nitrosomonas which can oxidize ammonia into nitrite in a process called nitrification. So nitrification is an important step in nitrogen fixation of the nitrogen biogeochemical cycle. And this is the following chemical reaction. 2 molecules of ammonia plus 3 molecules of oxygen gives rise to, in the presence of nitrogenase, gives rise to 2 molecules of nitrite, 2 protons and 2 molecules of water. Here Nitrosomonas derives energy from the oxidation of ammonia, an inorganic compound, and typically uses carbon dioxide as a carbon source. This process is common in soil and aquatic environments supporting the nitrogen cycle. So as I mentioned earlier before, in aphotic zones and in the deep pond environments where light is less and nitrogen ammonia oxidation yields energy using other chemicals as an energy source, and CO2 is used as a carbon source by Nitrosomonas. So if we refer to the overall nitrification reaction, ammonium is oxidized to nitrate. Ammonium is oxidized into nitrite, which is NO2 minus, by Nitrosomonas by ammonium monooxygenase, and Nitrobacter species nitrite oxidoreductase oxidizes it further to nitrate. So ammonia to nitrate is then referred to as the nitrification process. Now here is a comparison between different classifications of organisms: autotrophs, chemotroph, phototroph and heterotroph. And here is the comparison between chemolithotrophs versus chemoautotrophs. So depending on the energy source, the organisms can be divided into photo or chemo, where they use sunlight as a source of energy, which are called phototrophs, or breaking chemical bonds, which are called chemotrophs, right? Electron donor source can be organic, then it is photo-organoheterotroph or photo-organoautotroph, and inorganic photolithoheterotroph. Depending on the carbon source, again they can be further classified. Autotrophs use CO2 as the carbon source, and photolithoautotrophs use sunlight as the energy source, inorganic electron donor source as CO2 as the carbon source. So Nitrosomonas or the lithotroph is a chemolithoautotroph. This uses CO2 as a carbon source, inorganic electron donor source, but is a chemo as an energy source, use chemicals as energy source like ammonia. And here is a comparison between chemolithotrophs and chemoautotrophs, organisms that use chemical energy from inorganic compounds to synthesize all the compounds, while an organism, typically bacteria, which derives energy from oxidation of inorganic and organic compounds, use inorganic molecules at their final electron acceptor use oxygen as their final electron acceptor. Sulfur oxidizer, nitrifying bacteria, iron oxidizer, and extremophiles here that live in hostile environments such as deep sea vents. These are examples of chemolithotrophs versus chemoautotrophs. So lithotrophs are again the ones which use chemicals as energy source. And the example is ammonia oxidizing bacteria, Nitrosomonas. You guys have answered very correctly. I hope it is clear to everyone. Let me know in the chat and we will move on to the next question. Okay, I think we should move on to the next question. Question number 4. Question number 4 is asking us to define facultative anaerobes. Facultative anaerobes, option A, cannot survive without oxygen. Option B, are killed by oxygen exposure. Option C, grow better with oxygen. Option D, grow better without oxygen. Option E, do not need oxygen but are not harmed by it. Facultative anaerobes. Which is the correct characteristic suiting facultative anaerobes? Let us take a minute or so and collect some answers in the chat box, and I'll come back to this question. Okay, it's enough time for this question. Facultative anaerobes. Let us check the answers in the chat box. Divya has answered option E, Ashwini C, and Saida is saying option C. Let us see which of these description fit facultative anaerobes. The correct answer is option C, grow better with oxygen. Option C is the correct answer. Facultative anaerobes grow better with oxygen. So Ashwini and Saida have answered correctly. Well done. And Divya, good luck next time. Now, what are facultative anaerobes? A facultative anaerobe is an organism that can switch between aerobic respiration without using any oxygen, using oxygen, and anaerobic respiration without oxygen, depending on the availability of oxygen. This adaptability allows them to thrive in both oxygen rich and oxygen poor environments. Such as E. coli, found in the human gut, is a well known facultative anaerobe. In the presence of oxygen, it uses aerobic respiration. When oxygen is scarce, such as in the lower intestines, it switches to anaerobic respiration using alternative electron acceptors like nitrate or fermenting sugars to produce energy. And this is a distribution of uh, different microbes classification from low to high oxygen levels. For facultative aerobes use oxygen but can survive without it. Similarly, for facultative anaerobe, use they also facultative anaerobe can grow better in in presence of oxygen, can grow better because they are facultative anaerobe. Facultative means when condition arises, they are an anaerobe. So naturally, they are aerobes. I hope you understand this naming nomenclature. Facultative and obligate, two terms, right? Facultative means whenever there is a situation or depending on a condition, they can change their characteristic. So the microbe or the definition that we were talking about is a facultative anaerobe. Only when there is an adverse condition, it can become a facultative anaerobe. But it is generally an aerobe. It is generally an aerobic bacteria, but only when there is a condition, it will become a facultative anaerobic bacteria. Therefore, it grows better in presence of oxygen, but can also utilize anaerobic respiration for its availability. But it is usually an aerobic bacteria. Only when there is a condition or only when there is low oxygen levels, it can facultatively change into an anaerobe. While on the other hand, what does obligate means? Obligate means it's absolutely necessary. An obligate aerobe will only survive in presence of oxygen. It will die when there is no oxygen. Same goes for obligate anaerobe. It is unable to use oxygen and can't survive if it is present. So that is a subtle difference that comes between these definitions, and that is why option C is the correct answer. Facultative anaerobes grow better with oxygen, can also switch to anaerobic respiration when there is low oxygen. I hope you guys are clear about the definition. Two terms, facultative and obligate, and how they are used. Facultative only depending on a condition if the characteristic changes, and obligate is absolutely necessary for this condition, otherwise the bacteria won't survive. So an obligate anaerobe only will survive in absence of oxygen, while a facultative anaerobe will grow better in the presence of oxygen. But if oxygen is not present, then it's also okay, it can switch to anaerobic respiration. I hope it is clear to the students. Let me know in the chat. It's a very common mistake people make. You have to be very clear about facultative and obligate when you are classifying organisms based on the oxygen dependency. Okay, very good. Thank you, Divya. Moving on to the next question. Question number 5. Nitrogen fixing cysts formation is an example of dashed. Nitrogen fixing cyst formation is an example of dashed. Microbial growth, option A. Option B, microbial proliferation. Option C, microbial differentiation. Option D, endospore formation. Option C, E, exospore formation. Which of these can be described best for nitrogen fixing cyst formation? For a hint, it is a nitrogen forming cyst called heterocyst in nitrogen fixer Anabena. Let us take a minute or so and collect some answers in the chat box, as well as those who will be watching this video. Okay, enough time for this question. Nitrogen fixing cysts formation is an example of dashed, and in the chat, Saida and Ashwini have answered option C. Okay, let us see if it is the correct answer or not. And it is the correct answer. Option C is the correct answer. Nitrogen fixing cyst formation is an example of microbial differentiation, and all other options are not appropriate. Now, what happens in a cyanobacteria like Anabena? They undergo microbial differentiation to optimize nitrogen fixation and form such cysts known as heterocysts. Now, in a heterocyst in cyanobacteria like Anabena, certain cells differentiate into heterocysts under nitrogen limited condition. So, whenever there is a scarcity of nitrogen or low nitrogen levels, some cells in the Anabena, which is a chain like microbe, some of these cells differentiate into what is known as heterocysts. These specialized cells provide low oxygen environment necessary for the oxygen sensitive nitrogenase enzyme to fix nitrogen. Yes, this is important. This heterocyst creates a low oxygen environment so that nitrogenase, which is the primary enzyme for fixing nitrogen, which is also very sensitive to oxygen, can require only anaerobic conditions to function, can function without oxygen in a low oxygen environment inside the heterocyst. Heterocysts also lack photosystem 2, which produces oxygen and rely on adjacent vegetative cells for nutrients. So the heterocyst's sole job is to fix nitrogen. Nutrition is provided by its neighboring vegetative cells. So this is how a heterocyst development looks like. After 24 hours of the Anabena, the heterocyst develops. Some of the cells in the Anabena differentiate into heterocyst under nitrogen limited concentration, and these become the hotspot for nitrogen fixation, while other cells, the vegetative cells, provide nutrition to these kind of cells. And inside the heterocyst, with the adjacent vegetative cells. Sucrose is going through as a nutrition source to this heterocyst, and the presence of the nitrogenase enzyme here, which is fixing atmospheric nitrogen into ammonia. Now, what is an endosphere and endosphere and the exospore? Sorry, endospore versus the exospore, which was the fifth option. Endospore present within the cell, the spore in the cell wall, and exospore is a spore present outside the cell. Endospore is a structure found by bacteria to survive under unfavorable environmental conditions, consisting of DNA and a small amount of cytoplasm, while exospore is an asexual spore that is separated from the mother cell by the formation of a septum. So, endospore is a protection structure for coagulating or for binding DNA and small amount of cytoplasm, tightly packing the DNA and saving it from environmental damage, while exospore is a reproductive asexual spore that is separated from the mother cell or the parent cell. Endospore is produced by bacteria, while exospore is produced by fungus or algae mostly. Endospore is produced inside the mother cell, while exospore is produced near an end of the mother cell. Produced by cell division of the mother cell, not involved in the formation of endospores. The cell division is not involved in endospore formation. Released by rupturing the mother cell, released by budding. So the exospore can just bud out from the mother cell, does not need rupturing of the cell. Only one can be produced per organism, the endospore, only one can be produced. Whereas for exospore, several can be produced for reproductive potential of the organism. Produced by bacteria, genera Bacillus, Clostridium, and Penibacillus, while exospore is produced by conidiospores, Chamaesyphon, Psychosiphon, Stichosiphon, Actinomyces, and all other fungal species. I hope it is clear to the students that how heterocysts are formed by microbial differentiation of the existing cells in an anabaena in low nitrogen containing environment due to low nitrogen atmosphere, and inside the heterocyst is a low oxygen environment to promote nitrogenase activity of nitrogen fixation. I hope it is clear to the students. Just let me know in the chat box. I guess it is, since people have answered it correctly. Saida and Ashwini. Good. Thank you. Now here is the sixth question of this session. Mushrooms are dashed. Which definition fits the mushrooms perfectly? Mushrooms are option A, photosynthetic parts of underground multicellular fungi. Option B, fruiting bodies of underground fungi. Option C, mutualistic associations of algae and fungus. Option D, aggregates of protists. Option E, aggregates of helminths. What are mushrooms? Any definition from these options if it fits the characteristic of mushroom. Okay, I think it's time for this question. Enough time. Let us check the comments. Ashwini has answered option B. Okay, let us see what is the correct answer to this question. Mushrooms are fruiting bodies of underground fungi. Ashwini got it right again. Mushrooms are the fruiting bodies of fungus. So, dissecting the structure of a mushroom or the fungus, we have what is known as mycelium and the fruiting body. Mycelium is the underground web like root of the fungus. How the mushroom gets its nutrients and connects with the world around it. The mycelium is responsible for nutrient absorption and connection of the mushroom with the environment. Just like the root of any plant, the mycelium is a root of the fungi. Low in beta glucans and high in starch with no nutritional value. From a perspective of nutritional value, mycelium increases the surface area of the fungus and its major job is absorption of the fungus. Fruiting body, the classic recognizable cap and stem of the mushroom is the fruiting body where the nutrition value is stored and it is the reproductive structure of the mushroom. A natural tried and true method for extraction of the fruiting body. Mushrooms are the reproductive structures of certain fungi, specifically the fruiting bodies that emerge from the underground mycelium. And looking at all of the other options, photosynthetic parts of underground fungi, this is incorrect because fungi are not photosynthetic. They lack chlorophyll and cannot produce energy from sunlight. Photosynthesis is characteristic of plants and only some algae. Fungi are also not green in color. They are mostly whitish to brown to red in color. And mushrooms are the reproductive structures of the fruiting bodies of the fungus. Option C, which was also not the correct one, mutualistic associations of algae and fungi. These are known as lichens. This refers to lichens which are symbiotic relationship between fungi and algae or cyanobacteria. While some fungi form mutualistic relationship, mushrooms specifically are not defined by this association. So, mutualism between algae and fungi creates a different species called lichens. So, comparison between fungi and lichens. Fungi is composed solely of the fungal cells, heterotrophic and obtaining energy from the organic matter found in soil, water, and dead and decaying matter. Mushrooms are often found in dead and decaying wood, over leaves, and wet barks, absorbing nutrients from the organic or allochthonous matter. Lichens Lichens are the symbiotic association of fungi and algae. Obtain nutrients through symbiosis, fungi from algae cyanobacteria. Found on surface like rocks and soil in harsh environments. Lichens are often found in tundra ecosystems. So the cyanobacteria fixes atmospheric carbon dioxide and nitrogen for the fungi, and fungi provides harbor, support, and home to the cyanobacteria. So it's a mutualistic symbiotic organization association between a fungi and then cyanobacteria or algae. Aggregate of protists. Mushroom is never a protist. Protists are diverse group of eukaryotic microbes, and mushrooms are not aggregates. So here is an example of a paramecia, which is a protist of the protist family, and helminths are again parasitic worms, and mushrooms have no connection to them. Examples of helminths I have mentioned here: roundworms, liver flukes, tapeworms, and thorn-headed worms. So these are very different things from fungi, and mushrooms are the fruiting bodies or reproductive structures of the fungi, which helps in dispersal of the spores of the fungus. While mycelium is the root of the fungus which absorbs most nutrients and act to increase the surface area while connecting the mushroom to other mushrooms. Okay, is it clear to the students since you have answered everything correctly? Quickly let me know in the chat box and we can move ahead with the next question. I guess we should be moving ahead. Question number 7. Which of the following organisms are beneficial for wastewater treatment? Which of the following organisms are beneficial for wastewater treatment? Option A, helminths. Option B, protozoa. Option C, fungi. Option D, cysts. Option E, amoeba. Which of the following are beneficial for wastewater treatment? Are they helminths, protozoa, fungi, cysts, or amoeba? Let us take a minute or so to collect some answers in the chat. And for those who are watching this recorded video, please think of an answer. Okay, I think it's enough time for this question. Which of the following organisms are beneficial for wastewater treatment? Let us see what are the answers in the chat box. Only Ashwini has answered option C. The correct answer to this question is option C, fungus. Fungi are beneficial for wastewater treatment, and they have opened up a new era of treating wastewater with their beneficial properties because of their characteristic functions. Now, fungi are beneficial in wastewater treatment because they can degrade complex pollutants like lignin, certain chemical dyes, plastic, and even hydrocarbons. So, fungi have diverse set of organisms and enzymes which can degrade such complex pollutants in the wastewater. Another another characteristic of fungi is that they can survive in harsh and contaminated environments. So fungus already survives on dead and decaying matter. They can also survive in harsh environments where other microbes may not. Removing heavy metals through bioabsorption and transformation. Many fungus are known to absorb heavy metals and thus decontaminate the water before dropping it into the ocean or in rivers. Work synergistically with bacteria. Fungi also form many association with other organisms. The a beautiful organized association with algae was seen in the previous example where it formed a lichen. And fungi also provide eco-friendly and cost-effective bioremediation. So what makes fungi so durable and such a good choice for wastewater treatment? Enzyme production. Fungi releases extracellular enzymes including ligninases, cellulases, and peroxidases. So it can degrade lignin, which is a polymer, and cellulases, the cell walls of plants and other bacteria, as well as peroxidases, which can break down major pollutants. These enzymes play a key role in breaking down complex organic pollutants into simpler compounds. So one of the key feature of fungus is the variety or repertoire of enzyme production. Biofilm formation. Fungi form biofilms on surfaces creating microenvironments that enhance their attachment. So owing to point number 2 where they can survive in harsh and contaminated environments, because of biofilm formation they secrete extracellular matrices and enzymes and other factors which help in recruiting more spores and fungus community to form a rigid biofilm which enhance their attachment, which is essential for wastewater treatment. Biofilms provide a matrix for absorption of contaminants, contributing to treatment efficiency. So also biofilm harboring these mycelia and the fungus increases the surface area drastically, aiding in the adsorption of many contaminants from the wastewater. Nutrient assimilation. Fungi assimilate and transform nutrients including nitrogen, phosphorus from the wastewater. So fungi are also able to fix nitrogen and phosphorus, many species of fungi from the wastewater. Therefore, reabsorption of nutrient and assimilation. Nutrient assimilation reduces nutrient concentration in the treated effluent. So now the water which was generally rich in nitrogen and phosphorus is absorbed by the fungus, and this water will be less likely to be causing eutrophication in the recipient water body. Contaminant removal. Fungi contribute to the removal of contaminants through various processes. This includes absorption, degradation, and transformation of pollutants, leading to cleaner wastewater. So, absorption of heavy metals, transformation of wastewater, and various processes, other processes, surface adsorption by the fungi can lead to cleaner wastewater as an effluent being transported into the rivers. That is why fungi are now a good choice for beneficial waste treatment because of these properties. So here is a schematic showing the process of using fungi for bioremediation process, now termed as mycoremediation, in which fungi can absorb oil, hydrocarbon waste and heavy metals, digest and metabolize the waste, and conversion of water into harmless gases. So all this role can be performed by fungi, not just this but other diverse roles like accumulation of aquatic pollutants like waste disposal, agrochemicals, industrial, atmospheric deposition and mining activities. All these things can be absorbed through the porous mycelia of the fungi and present in biofilms. And in biosynthetic pathways, such components of the fungus can be reused and recycled for precipitation of surfactant, desorption mechanism of pollutants, interface of soil water pollutants, and biosurfactant production. So these metabolites or chemicals which are adsorbed onto the fungal surface can be downstream processed for biosurfactant production, and this is how fungi can be used for mycoremediation process, not only refining wastewater but also absorbing heavy metals and nutrients and pollutants back and utilizing them for manual uses and for biosynthetic pathways. That is why fungus is now a good choice for bioremediation, owing its name to mycoremediation because of the large repertoire of enzyme production, biofilm capability, assimilation of heavy metals and nutrients, and contaminant removal. I hope it is clear to the students that the uh, fungus is such a great microbe and an organism which is helping us in clearing the wastewater treatment. If it's clear, then let me know in the chat box and we can move to the next question. Okay, I think it is clear to everyone. Moving on to the next question, question number 8. Which of the following is not an essential micronutrient for microorganisms? This is an information based question. Micronutrients present in microbes. Which of the following is not an essential micronutrient for microorganisms? Option A, W or wolfram, which is tungsten. Option B, V or vanadium. Option C, Se, selenium. Option D, As, arsenic. Option E, Cu or copper, cuprium. Which of the following is not an essential micronutrient for microorganisms? Let me know what you think
of the answer in the chat box, and I'll be back to this question in a minute. Okay, I think it's enough time for this question. Which of the following is not an essential micronutrient for microorganisms? And Ashwini has answered arsenic. No other answers? Okay. Let us see which is the correct answer. Option D, arsenic, is the correct answer. Well done, Ashwini. Arsenic is not an essential micronutrient for microorganisms, but all other elements are required in some or the other microorganism. Let us see the examples of W or tungsten, wolfram, essential in some microbes, used in enzymes called tungstoenzymes, in which such as formate dehydrogenase and aldehyde ferredoxin, oxidoreductase, in hyperthermophilic archaea like Pyrococcus furiosus. Tungsten containing oxidoreductases are vital for metabolism in methanogens. So, tungsten containing enzymes are mostly found in archaea and methanogens, extremophiles. So, most of these metals or micronutrients act as coenzymes or cofactors for enzyme activity and function. Vanadium, essential in some microbes present in vanadium dependent nitrogenases. We know that manganese is the common uh, coordination metal in many nitrogenases, but there are other different nitrogenases which are vanadium dependent in certain nitrogen fixing bacteria, like allows microbes to fix nitrogen when molybdenum is scarce. Example, azotobacter can use vanadium nitrogenase for nitrogen fixation. Halomonas species can respire arsenate, but this is not a general microbial requirement. selenium Se is essential. Selenocysteine, the 21st uncommon amino acid, the 21st amino acid, important for formate dehydrogenase and glycine reductase in anaerobic bacteria. E. coli requires selenium for formate dehydrogenase activity. Arsenic is not essential. It is generally toxic to microorganisms as well as humans. Some bacteria can use arsenate or arsenite in detoxification or respiration, but it is not required for normal growth. Unlike wolfram, vanadium, selenium, or copper, arsenic is not a universal micronutrient. So that is why arsenic is the correct answer. It is toxic. Copper is an essential cofactor in cytochrome C oxidase, superoxide dismutase, and many other important enzymes in the electron transport system. Example, Pseudomonas uses copper in multi-copper oxidases for respiration. And copper is an essential micronutrient. So that is why arsenic is the correct answer. I hope there is no problem in this question that why arsenic is the correct answer. It is toxic to the microorganisms and it is not a general universal micronutrient. Some bacteria can use it in detoxification or respiration, but it is not required for normal growth. Okay, moving on to the next question. Question number 9. Two more questions and we will be good to go to end this session. Question number 9. Which of the following statements is true? Option A. Proteins are the most abundant macromolecules in any cell. Option B. Glycogen is used for storage only by eukaryotes. Option C. All components of a complex medium are precisely quantified components. Option D. Defined medium can show batch to batch variation. Option E. Storage compound PHB can be formed during oxygen starvation. Which of these is a truth statement? Read all the options carefully and let me know what do you think is the correct answer. Proteins are the most abundant macromolecules in any cell. Glycogen used for storage only by eukaryotes. All components of a complex medium are precisely quantified components. Defined medium can show batch to batch variation. Storage components PHB can be formed during oxygen starvation. I think it's enough time for this question. Time to reveal the correct answer. Let me check the chat box if there is some answer. Ashwini says all, sir. Okay. I think only one statement is true, which is option E. Storage compound PHB can be formed during oxygen starvation. This is the true statement of all options. Let us verify each option.
Of course, this is a MCQ and all options might not be the correct answer, Ashwini. Let us go option by option and analyze everything until we reach the final one. Okay? Now, what is PHB? Polyhydroxybutyrate or PHB is a biopolymer synthesized by bacteria and archaea as a storage compound for carbon and energy. It's accumulated when these microorganisms encounter a surplus of carbon or experience nutrient limitations. PHB is essentially a bacterial fat or energy reserve similar to how humans store energy as fat. So PHB is nothing but equivalent to the adipocytes which are present in humans. It is a biopolymer which is a storage compound for carbon and energy. Biopolymer of carbon, whenever there is surplus of carbon or any nutrient scarcity, PHB bodies can be seen inside certain bacteria. So there are PHA polymerase which are polyhydroxy and PHA depolymerase, and they make this polymer PHB, and there are such big granules of PHB visible inside a bacteria when there is carbon surplus or food surplus. And this can be also produced in oxygen starvation conditions for storing carbon as an energy source. See, polyhydroxybutyrate is a carbon energy storage polymer in many bacteria, accumulates under nutrient limitations such as nitrogen, phosphorus or oxygen starvation with excess carbon source. Whenever there is a nutrient limitation but there is excess carbon source, such bodies can form, helps bacteria survive stress and resume growth when conditions improve. Looking at all other options, proteins are most abundant macromolecules, not true universally in many microbial cells, especially RNA. rRNA is actually more abundant than proteins, 20% versus 55% dry weight proteins, but RNA outweighs DNA. The wording any cell makes this false. So it is not in any cell that the protein is the most abundant macromolecule. It can be ribosomal RNA, because in actively translating cells and in many other cells, rRNA outweighs proteins, so it is not true universally. Glycogen is used for storage only by eukaryotes. Wrong, because many bacteria like E. coli, cyanobacteria also store glycogen. It is not limited to eukaryotes. Option C, all components of a complex medium are precisely quantified components. This is also incorrect. Complex medium does not contain chemically defined components. Contains ingredients, natural ingredients like yeast extract, peptone, beef extract, whereas defined medium contains all components uh, quantified perfectly. Defined medium can show batch to batch variation. Again, going on by the definition, defined media are consistent since chemicals are weighed exactly. It is complex media that show batch to batch variation due to natural extracts present in the complex media. And PHB, which is polyhydroxybutyrate, is a carbon energy storage polymer which, when there is oxygen starvation as well, there can and carbon surplus, there can be formation of PHB bodies inside the bacteria, which helps bacteria as a stress survival mechanism. I hope it is clear to everyone that PHB is a storage response as a response of stress to bacteria whenever there is nutrient scarcity but carbon surplus or energy surplus. The bacteria will tend to store this as fat cells. These are the fat cells of bacteria, the PHB bodies. While all other statements might seem to be true, there are little catches present in the statement which makes only option E the correct statement. Okay, moving on to the final question, question number 10.
Which of the following statements correctly describes the respective cell cycle stage? Option A, G1 begins after DNA replication is completed. M phase is a period between G1 and G2. S phase is a period of DNA replication. G2 is followed by S phase. G0 is the period between completion of cell division and initiation of DNA replication. Again, very closely related statements about the cell cycle, different phases of the cell cycle. You have to tell me which of the following statement correctly describes respective cell cycle stages. Now let us take some time and arrive at the correct answer in a minute or so. Question is about cell cycle phases. Okay, I think it's enough time for me to reveal the correct answer. Okay, Saida has answered option C, Ashwini has answered option C, and C is the correct answer, guys. S phase is a period of DNA replication, is the correct statement representing the phases of cell cycle. Well done. Final question, and we got some participation. Now, knowing all the phases of cell cycle is consisting of 5 phases, 4 active phases cell undergoes before cell division, right? So the first phase is gap phase 1 or G1 phases. Following cell division, cells in G1 grow and prepare for DNA synthesis. Notable proteins expressed in G1 include APCC, CDH1, cyclin D, CDK2, RB1 and everything. So G1 phase is the growth phase for the cell which has just divided. It will grow in size and prepare for DNA synthesis. For DNA replication, whatever proteins are necessary will be made in the G1 phase. S phase is the actual phase in which replication of DNA takes place, or DNA synthesis, such as and proteins such as MCM, PCNA, CDC45, these are all accessory proteins of the DNA replication machinery are activated, and they transition. Key proteins needed to transition into this phase include cyclins. So across the cell cycle there are two group of proteins, cyclins and cyclin dependent kinases, which maintain the tempo of the cell cycle. So many certain cyclins are stage specific and only get expressed, and the levels go up in certain cell cycles phases. So DNA synthesis occurs in the S phase or synthesis phase. The amount of cellular DNA which is doubled in G2M relative to G1 G0 can be used to distinguish cell cycle stages. Gap phase 2 or G2, after DNA synthesis, G2 serves as a preparatory phase before mitosis. So it is preparing the cell for mitosis, which is the actual division, and M phase is nothing but the mitosis phase. Mitosis consists of again several phases such as prophase, metaphase, anaphase and telophase. Okay, mitosis, there are several notable proteins that are required to initiate and complete the stages of mitosis, such as many cyclins and other metaphase related proteins, right?
while G0 phase is a quiescent phase. From the G1 phase, quiescent, senescent or differentiated cells can enter G0, where the cell is actively out of the cell cycle. It's a G0 phase or a quiescent phase where it is not dividing anymore. Diminished level of proteins such as Ki67 and FOXM1 are the hallmarks of this phase. So this constitutes all the phases of the cell cycle. G1 or growth phase, S or synthesis phase, G2 is preparatory phase for mitosis, and mitosis is the phase where actual cell division happens. G0 is the quiescent phase. I hope it is clear to the students. I think it is, and since you guys have answered correctly, so with this we have come to the end of our presentation. Solved 10 questions from week 4. I will go back to the meeting again. Stop my presentation. Yeah. So with this we have come to the end of this live session where we solved 10 example multiple choice questions from lecture week 4 from diverse topics. I hope you liked the class, and I'm happy that you guys are interacting well. The population is thin right now, but it doesn't matter. These are recorded classes and will be viewed by many students in the future. I hope you enjoyed the class, and if you have any suggestions for me or the way of presentation, you can let me know, or if you have any doubt. Else, I will end this call and see you soon next Saturday with week 5 lectures, and we'll be dealing with assignments of week 5. Thank you all for joining, and I'll end this call. Thank you.